in this video, I'm going to go through the genetics PowerPoint and just hit on the most important parts. So I'm going to skip over some slides that I don't think are as important as others, and I'm going to try and just hit on the most important things. So we're learning about genetics, and genetics is basically looking at heredity, or the passing on of characteristics from one generation to the other. So we're taking what we learned in cell reproduction through meiosis, and we're looking at how mom and dad made kids. Okay, and we're looking at how their traits got passed on to you and kind of why families all look the same or very similar to each other. The man we can thank for this unit is Gregor Mendel. He was an Austrian monk, and we call him the father of genetics because he is really the reason why we understand all of this stuff about genetics. He was a monk, so when you are a monk, you live in a monastery, and you usually you get some time to study something that interests you when you aren't studying the church. So he chose to be a botanist, and he worked out in a garden in a greenhouse, and he eventually started experimenting with pea plants, and he kept a journal, and he made a lot of conclusions that have been found to be very true and that we still believe today. So he's the father of genetics, and he and the pea plants just formed this entire great unit. And I'm actually going to skip over his experiments because they can be a little confusing, and that's not the important part of this. But I do want you to understand what it means to be a true breeding organism. Okay, if you think about like a dog breeder, for instance, like if you are a dog breeder who breeds golden retrievers, right? You usually have golden retriever dogs that you mate to make the puppies that are true golden retrievers. They're not mixes with different breeds. They're going to be full golden retrievers who produce true golden retrievers. That's exactly what this means. So when you have two organisms that only produce almost identical organisms, this is called true breeding or a purebred. Okay, so if you think of a dog breeder who breeds purebred labs or purebred German shepherds, that means that all of the babies of those German shepherds that they breed are going to be pure German shepherds. Okay, I'm going to skip through some of these. His experiment, basically what he did is with his pea plants, there's two different flower colors that pea plants could get. They can either be purple flowers or white flowers. So he took a purebred purple flowered plant, so a purple plant that only produced purple flowered plants, and he took a white plant or white flowered plant that only produced other white flowered plants, and he crossed them. He mated them and allowed them to reproduce, and he named that first generation of the purebred purple plant and the purebred white plant the P generation or the parental generation. So these are like mom and dad. Okay. And what he found was that the offspring, also known as the F1 generation or the first generation, so these are like mom and dad's kids, all of them were purple. They were all purple flowers. There was not a single white one. And he did this with thousands of different offspring. So he mated these many times and had many different babies, and 100% of them were purple flowers. Not a single one out of the thousands that he produced were white. Okay, so he decided that the purple flower trait must be dominant. It has to have masked any white trait that got passed down through flower color. Okay, and then what he did is he took two of these first generation purple flowered plants and he crossed those. Okay, so it's kind of like inbreeding, but it's plants, and he was a botanist and he needed to experiment, and I'm very thankful that he did, because the result was that the F2 generation or the second generation, so these are like the grandkids, 75% or 3 out of every 4 were purple. The other 25% was white. So that white flowered trait skipped a generation, and came back in this F2 generation. Okay, so he concluded that the dominant trait must mask a recessive one. So he was guessing that some of these purple flowers in the F1 generation had a white allele or a white trait in them, but it was masked because it also had the trait for the purple flower. And then when he had these cross, he found that that recessive trait came back. So it must mean that all of these, the 25% of the white flowered plants, must have not had that purple flowered trait in them, and that's why they were white. Okay, so the most important things to get out of this right now is the P generation are the parents or the parental generation. The F1 generation are their offspring, and the F2 generation is going to be like the grandkids or the offspring of the first generation. Okay. Now, the conclusions that he came up with, which we still believe to be true today, he said that for each trait, an individual has 2 copies of the gene. So thinking back to when we learned about chromosomes, right? We have 2 copies of every chromosome. We have 2 copies of all 23 chromosomes, which gives us a total of 46 chromosomes altogether. Now, of the 2 chromosomes, we learned when we were talking about meiosis that we get 1 copy from mom, 1 copy from dad, and that's where we get the 2 copies of that gene. And he said that there were different versions of the genes, which we call alleles. So for instance, in the pea plants, the flower color, there were different versions of it. You could either be purple flowered or white flowered. Okay, and every plant that was produced got 1 of those alleles from mom, 1 from dad, and depending what those alleles were, they either produced purple flowers or white flowers. Okay. Now we also determined that there is a dominant allele, which is the expressed form of the trait, and the recessive allele, which is the trait that's not expressed when it's paired with the dominant one. Okay. And we will get to the point real soon to help us figure out how we can represent these when we're doing our own genetic crosses on paper. We can't do it with plants, that would take years, but we can do a quick genetic cross on paper to figure out what the offspring could possibly look like. So dominant allele, think of like the purple flower trait, is going to be expressed whenever it is present, and the recessive allele is not going to be expressed if there is a dominant allele with it. Okay, the only way to have a recessive allele expressed is when it's with itself and there's no dominant allele present. That's how we got the flowers in that second generation. Okay, we also concluded that when our gametes are formed, and again we learn this in meiosis, the gene for a specific trait gets divided. So when this cell becomes or goes through meiosis to become the sperm, or this one goes through the meiosis to produce the eggs, we separate the alleles. So if this is your mom's cell, she got 1 of these little t's from her mom, and she got the other little t from her dad, and when she produces her eggs, she puts 1 of the little t into 50% of her eggs, and she puts the other t into the other 50%. So each allele or each gamete is going to carry 1 allele, and then when mom and dad make you, that's how you get your 2 alleles. Okay. Yes. Let's go on to genetics today. To represent dominant and recessive alleles, we use letters of the alphabet to express them. Okay. To represent a dominant allele, we're going to use a capital letter, right? And then to represent a recessive allele, we'll use a lowercase. So if you look at this diagram here, right, capital H is going to represent dominant, and a lowercase is going to represent recessive. Kind of think of like when you write a word and the first letter is capitalized, or if there is a capital letter in the word, it always goes to the front of the word. We never capitalize like the 3rd letter in, right? We always capitalize the first one. So if we capitalize the first one, if there's a capital letter in any of our crosses, that means that whatever that dominant trait is, it's overpowering the rest of the letters in that for that trait, okay? And we want to make sure when we're doing these crosses that we use letters that don't look the same as capital and lowercase, because it can be very difficult, maybe not so much for you to tell, but for me to tell which one is dominant, which one is recessive. Even if you write the capital P really large and the lowercase p really small, it's still sometimes difficult for me to know, like if you have two little p's together, if you mean for those to be two little p's or two big p's. So just out of all the 26 letters, I'm asking you to not use the 1, 2, 3, 4, 5, 6, 10 on the screen, because it's just too difficult for me to tell the difference. And if I can't tell the difference, I don't know if you understand what you're doing or not. So please pick a letter like H, right? The capital H and lowercase h look a little different. Therefore, I can tell if you have a capital H, that's the dominant allele, and a lowercase h would be your recessive one. Okay? Now there's some terminology that you need to understand. If an individual has two alleles of a gene that are the same, okay, we call them homozygous. So for instance, if we have two capital B's, these are the same allele, that would be homozygous. Or if we have two little b's, those are the same allele, that would also be classified as homozygous. Now we have terms that differentiate this homozygous from this homozygous. We have homozygous dominant for the two capital letters, and we have homozygous recessive for the two little ones. If it helps you remember the difference, the prefix homo means the same. So this means that we have two of the same allele and they are both dominant. Therefore, we have big R, big R. Homozygous, homo means the same, which means we have two alleles that are the same, and this one just so happens to be recessive, and that's how we get little r, little r. Now, if you have two alleles that are different, like a big B and a little b, we call that heterozygous, okay? The prefix hetero means different, so we have two different alleles, therefore we have one dominant and one recessive. In this case, when you have a heterozygous genotype, or two alleles that are different, that means that the dominant trait is going to be expressed. It's going to mask this little guy. This big R is overpowering. You aren't going to notice this one. Okay? Now there's two more words we need to understand before we get into actual problems. The first is a genotype, and I used this on the last slide just briefly, but a genotype is the genetic makeup of an individual, or the alleles. Okay? So think genotype, genetic makeup. And that's where we pull out these H words that we learned on the last slide. That's where you would tell me if it's homozygous dominant, heterozygous, or homozygous recessive. The opposite then is going to be the phenotype, which is the physical appearance of the trait. So you would take the homozygous dominant or the heterozygous or the homozygous recessive and tell me what the individual looks like because of this. So for instance, you can tell me that they have brown hair or purple flowers, or maybe they have black and white fur. Based on the genotype, you can then find the phenotype for each of the organisms. So in all the problems we do, the very end step is going to be to tell me the genotypes and phenotypes, or the possible ones, for the offspring of a certain cross. It's important that you understand the difference between these two words. So genotype, think genetic makeup. Phenotype, think physical appearance. And the last part before we get into any of the problems, we need to learn how to use something called a Punnett Square to help us, because you're going to get tired of drawing these, okay? This is just a diagram that helps us predict the expected outcome of a genetic cross. So we're going to use a Punnett Square. Think of like Battleship. That's exactly kind of how you do these crosses. This is the tool you're going to use. Now what happens is you take mom's gametes and you're going to put them above each of the columns, or you can put them by the rows, doesn't matter where you put them, but let's just say mom's gametes go up here. and dad's gametes are going to go here, right? And then we cross them to show what the possible offspring can be. Okay, and this will make more sense when we do an actual example problem with monohybrid crosses. Just know the gametes go above the columns for either mom or dad, and then the other one, the gametes for mom or dad that you didn't put up here, go along each of the rows. And then when you cross them, the possible genotypes for the offspring will be found on the inside of the boxes. Okay, so a 2x2 Punnett square just like this is going to be the typical Punnett square that we'll use, but there will be a cross at the very end of the unit that actually uses a 4x4 Punnett square, and we'll get to that eventually, but right now this is going to be your best friend. Okay, and before I get into any of the crosses, I'm going to stop and I'll make individual videos for each of the crosses, but this is the bulk of genetics right here in this video with all the terms that you need to know.